This is Pharmaphorum web editor Nicole Raleigh, and I am on site in Berlin for ESMO 2025. And with me today is Jane Lee, who is Senior Director of Pharma CDX and CRO Partnership, Clinical Next Generation Sequencing Business at Thermo Fisher Scientific, which is a life sciences and clinical research company that provides a wide range of analytical instruments, laboratory equipment, and services, offering solutions for sample and material characterization, chemical analysis, clinical diagnostics, and biological therapeutics manufacturing. Pleasure to speak with you today, Jane.
Nice to meet you too.
Before we begin properly, I was just wondering if you were able to provide viewers with a very brief introductory background to your work and the journey that has led you to doing what you do today with Thermo Fisher.
So I've been with the company for 25 years,
Oh,
believe
wow.
it or not. I've been with the next generation sequencing business since the beginning. So that's 2010 when we acquired Iron Torrent technology, which then became the clinical sequencing business. So after 15 years, I've been with the business. The past 10 years focused on companion diagnostics. So, we've seen tremendous development in NGS and CDX. I'm really grateful and blessed to be part of this journey in the past decade.
Thank you. So the company, I believe, recently received FDA approval for its oncomine DX target test as you say, companion diagnostic or CDX to identify patients who may be candidates for HERNEXEOS, also known as zongertinib in tablet form, a tyrosine kinase inhibitor or TKI developed by Boehringer Ingelheim. And indeed, viewers will be able to listen to the podcast with Boehringer separately. And that test, Jane, allows clinicians and pathologists to assess if non-small cell lung cancer tumors harbor human epidermal growth factor receptor 2 or HER2, ERBB2, TKD activating mutations. So I was wondering if you could summarize how this addresses unmet needs in this cancer.
So if you look at cancer, lung cancer is the second most common cancer type for men and women in the US. And historically, it has one of the lowest 5-year survival rate. So it's no surprise that we've seen a lot of, biomarker-driven clinical trials and drug development in the lung cancer space in the past decade. So if you think about, 10 years ago, it was just EGFR and ALK inhibitors, two biomarkers. And then in the past 10 years, we now have, ROS1, BRAF, KRAS, NTRK, MET, RET, EGFR exon 20 insertions. And lastly, HER2 is the latest biomarker. So the first HER2 ADC was approved by the FDA in 2022. So now we're really excited with our pharma partner, Boehringer Ingelheim's drug, is the second HER2 TKI approved in this indication to serve the unmet needs in lung cancer.
Thank you. So can you also explain how progress in precision oncology depends on close collaboration between pharma and diagnostic developers?
Right. So precision medicine, in a simple term, is really targeted therapy and the biomarker testing to identify patients for the targeted therapy. So if you think about 10 years ago, majority of the targeted therapy and the biomarker testing are single gene biomarker, single gene using PCR, FISH for molecular testing. That has completely changed in the past decade. As I mentioned, in the non-small cell lung cancer case, there are now 10 biomarkers, 20 targeted therapies for those across those 10 biomarkers in lung. So single gene testing is just not becoming feasible. So what we've seen in the past decade is NGS has really emerged as the preferred technology for biomarker testing because it can identify hundreds of genes at the same time from the cell tissue, and the plasma. So, if I think about a decade ago, most pharma collaborate with fish technology diagnostic companies or PCR diagnostic companies. Um, and now in the past decade, you know, most pharma are collaborating with NGS companies.
Mm.
So we're very fortunate to be part of that journey.
That's quite a journey.
It is.
Indeed, yes. So my next question is how, through co-developing companion diagnostics with targeted therapies and aligning early with regulators, do these partnerships that we've been discussing ensure accurate patient drug matching, as well as faster approvals and greater global access to breakthrough treatments?
Mhm. So for targeted therapies, in many countries, there is a specific regulations that require companion diagnostic as part of the new targeted therapy submission approval through the regulatory pathway. So as an example, in US, the FDA requires contemporaneous submission of CDX and new targeted therapy and approval. In Japan, the PMDA also requires a contemporaneous launch process between the CDX and the new targeted therapy. And then the new IVDR regulation, CDX is also included as part of the drug submission to EMA. And then this trend is kind of spreading. So if we look at other countries like Australia and Korea, there is also policies shifting to recommending CDX with targeted therapy. So I think what that means is for a drug company, as you develop targeted therapy, you need to have a global mindset.
Mm.
And then you need to have an understanding of the different regulations and pathways in different countries and understand the timeline implication. And this is the most important that we see with some of the pharma clients. They think about CDX too late.
Mm.
And then they would come to a diagnostic company to develop CDX, but realizing the CDX development takes time. And then that time needs to match with the drug timeline. So we always encourage our clients, when you have a drug in development, think about CDX and put on your radar, start planning for that sooner rather than later, because it takes time for that co-submission process.
Thank you for those insights. So additionally, Thermo Fisher Scientific announced that the oncomine comprehensive assay plus is now available on the ion torrent Genexus system, helping accelerate precision oncology research. And that oncomine comprehensive assay plus detects a broad range of genomic alterations in 517 genes, and will now be able to deliver comprehensive genomic profiling, also known as CGP, results as soon as the next day. That's incredible.
That is.
And I believe that's enabled by ion torrent technology and easy automated workflows. So please tell us more about this.
Okay, love to. So comprehensive genomic profiling, or CGP, is becoming quite a focus for in cancer research.
Mm-hmm.
So in cancer research, you have what we call targeted panel, which are the smaller panel typically, and they can identify most of the oncodriver mutations. But you have CGP that they typically look at 500 genes, including complex signature like TNB, MSI, and HRD. So this is the trend that we see, that a lot of the targeted therapies, they started with simple variant class, but they're moving towards complex signature like TNB, MSI, and HRD. So the value of the CGP is you can gather all the insights broadly from the same panel. However, CGP is not a new concept to the market per se. It was introduced to the market several years ago. However, the key challenge with CGP is because it's 500 genes, it takes too long to get the results back, and it requires a lot of expertise. So it's the use often is limited to leading medical centers or cancer centers where you have PhDs, really sophisticated user to interpret the results. So our go with the Genexus platform, which is the first of a kind that's automated NGS workflow. So if you think about it, is an average laboratory, smaller laboratory with a non-NGS experts users can use the instrument because it's automated, so you can get the results back. So this platform was launched into the market several years ago and had a lot of enthusiasm from customers because all of a sudden, smaller labs who didn't feel comfortable using NGS, all of a sudden now they see a way to deploy NGS in their smaller labs. And then the other piece is the GeneXus platform is really rapid. It's the first rapid NGS system on the market that can deliver results next day. So we know that, in cancer, we always say time is life. Speed is everything when you have a patient waiting for treatment. So the oncomine comprehensive plus assay is the first time a CGP 500 gene panel can generate results next day and can also be deployed into smaller labs outside these leading medical centers, can be used by non-NGS users. So, the significance of this product launch is it really broadened the NGS access to the market, to broader users, and to broader countries where they may not be having sophisticated technicians to operate NGS.
Brings a new nuance to the expression speed is of the essence, certainly.
Yes.
Right, so let's sort of tunnel down slightly more. And how is NGS, and if you need reminding viewers, which you probably don't, but next generation sequencing, how is that driving the future of, importantly, personalized medicine, helping to bring targeted therapies to patients faster?
Right. So, NGS had an extraordinary innovation in the past 10 years. So you now have small panel, big panel, NGS solution from tissue, NGS solution from liquid biopsy, NGS for targeted therapy, for MRD. So there is an explosion of NGS applications in different sample types, different disease indications and all of that. However, I think one key challenge still remains that NGS is just too complex and not fast enough. So all this innovation can only be used by expert users. So you can see it limits, who and the locations where it can generate benefits to the patient. So for us, a key focus, that we want to enable NGS in the next couple of years is to really broaden the access to make it equitable care. Right. So, if a patient, if you're living in an emerging country, you're in a small hospital, you can have access to NGS solution and targeted therapy. So it's not just a small group of patients closer to medical centers that would benefit. So I think for us, we really care about this equitable care. And in order to achieve that, you really need to have the NGS that's simple and easy to use everywhere and that can be deployed anywhere and fast.
Like GeneXus.
Like GeneXus. Yes.
Indeed. Okay, so my sort of final question, as it were, is what do you think, given everything that's being discussed here at ESMO 2025, what do you think is going to be, obviously speaking more broadly now, the sort of key takeaway, and what will that mean for the future of oncological care?
Yeah. So, today is second day at ESMO. This is my 10th year attending ESMO. I continue to be fascinated on the rapid development of the therapeutic landscape. So now you have chemo, targeted therapy, immunotherapy, ADCs, variety of different therapeutics with different biomarkers, some molecular, some protein. And then you also have now biomarkers. They start with one indication, and now they're becoming cancer. So I think there is tremendous pipeline and a lot of new therapies, new biomarkers to come. So NGS, I think, is going to be mainstream in the future. And I think NGS is going to expand its usage, maybe along the entire patient journey. So, not just in therapy selection, but maybe MRD, and not just in tissue and blood, but also maybe in urine samples. So there's a lot of expansion as these therapeutic targets look at new biomarkers, and the NGS adapt to, you know, um, to basically become the solution for those biomarkers.
Thank you very much for your time today, Jane. It's been a pleasure.
Likewise. Thank you.